Thank you very much, Roy, for agreeing to give a Build a Cell seminar, and you can get started.
Okay, hello everyone out there. My name is Roy Bar-Ziv. I'm a professor at the Weizmann Institute in the Department of Chemical and Biological Physics. My background is soft condensed matter physics, and then at the level of a PhD, and then I did a postdoc at the Rockefeller where I transitioned to biophysics. Now I'm back for about 20 years now at the Weizmann, developing sort of two-dimensional artificial cells in 2D, something which really combines some of the ideas that are borrowed from soft matter physics and material science, together with biochemistry, molecular biology, and try to bring sort of new concepts. I see this as everything we do is foremost basic science driven, curiosity driven. And that's why I put up this slide to sort of review what we all know, but maybe just to put a focus on what I may think of things somewhat differently. To build an artificial cell, for many of us, I guess, is to understand the fundamentals of life by building. And the question is, what is a cell? Has probably many basic definitions, but we would all agree that, would have a membrane-bound unit which contains the molecules of life, capable of maintaining homeostasis, i.e. self-regulate to maintain dynamic equilibrium, to reproduce, grow, replicate the DNA and divide, and to carry out metabolism, communication, and evolution. So that's, the definition of a cell. What would be an artificial cell here? The natural course of action in science would be to define a cell as something which mimics, a real living cell, based on a membrane compartment with the DNA inside and the minimal components of life inside to kickstart a cell bottom up. And that's what many groups in the world nowadays work on to try to tackle the various problems associated with that. I had done my PhD on dynamic instabilities of membranes in various topologies and geometries, and I was not ignorant of that, but I nonetheless chose, when I started my lab here, to focus on an entirely, let's call it embodiment of an artificial cell, quote unquote. It's not a cell per se, it's a miniaturized compartment carved into a silicon surface, a wafer. And this is what you see here. This is our latest version of a compartment, which is basically a pancake, 2 microns deep, about 30 microns wide, with in the middle of which we, instead of having a plasmid or any other genome scale DNA piece floating around in the chamber when we close it, but actually we immobilize DNA polymers chopped into sort of individual genes, densely packed on the surface, acting as sort of a synthetic DNA program, if you think about genetics, or a reaction center. If you think about chemistry and catalysis, you see where this comes from. There is this capillary which enables free diffusion in and out through the well, going down through the wafer in this case, to enable free exchange of material, except for DNA, which is immobilized. Everything else is free to diffuse in and out. And you see where this kicks in to basically enable us to generate gene expression in steady state. So for me, an artificial cell, quote unquote, will be this sort of miniaturized DNA compartment carved into a solid state surface. And let's see where this has taken us. Now, to think, we use, like the rest of the community, making proteins in a cell-free system. The basic premise is to make proteins from DNA to RNA and to protein. There are two kinds of systems we all use, either the minimal system, i.e. the pure system invented by Shimizu and Ueda back 20 years ago or so. And here, this is a system which comprises about 100 components, including the ribosomes, tRNA, translation proteins, etc. And what I'd like to emphasize is gene expression reactions, transcription, translation are molecular machines that consume energy. And this is a far from equilibrium system composed of many components. So the far from equilibrium will come in shortly when we start to do these reactions under these confined conditions. Of course, the other systems that are nowadays used is revisited extracts, mostly based on bacteria, E. coli, which enable us to basically use the entire endogenous protein machinery and content of bacteria for regulation and so on. Our journey has basically taken us from 2003, where Vincent O'Rourke and I were postdocs at Rockefeller at the same time. This was the first attempt to basically reconstitute gene regulation circuit elements in a test tube reaction. That was the first naive approach to sort of show that you can mimic the lac operon or aspects of that by allosteric regulation of gene expression. It mostly showed us what are the bottlenecks and limitations of trying to go from a single gene to a more complex system of multi-genes and eventually to an entire cell. And this is where my conclusion was to start a lab and basically focus on two-dimension, where you have the spatial components and the material science implemented rather than in a vesicle, but in sort of quasi 2D. And we now have a real, a program where along the axis of biological complexity here annotated in number of genes, we've touched each and every aspect of this axis from 1 or 2 genes coding for regulation to a network comprised of 5 to 10 genes to machines or machine components, all the way to dozens of genes and proteins, all the way to actually show first step storage an autonomous cell capable of self-regenerating its machinery. We're working towards that. And here we're at the level of 100 or so and more genes and proteins, all the way to multicellular systems where we now have communication between such compartments that I'll show you shortly. So this is the advantage or the capabilities which have been enabled by using this two-dimensional DNA compartments. The general aims of our work is basically to understand biological self-organization and computation. And computation is an elusive concept in my view. Computation is something that we understand well with digital and analog, electronics and Boolean math and so on. But to actually define biological computation and try to implement that, I think is a big challenge, because one deals with concentrations of various components, and we don't yet have a clear theory how a synthetic system that can be programmed can actually do computation, and what is the scope of this computation. I think this is something that as a community we have to push forward in basic principles of combining self-organization of molecules that are being expressed to create computation. I'll show you one aspect of computation, the simplest one, which is decision-making. This whole approach basically simplifies, those complex and entangled processes in organisms across many scales, and I'll show you a few examples of that. Ultimately, we'd like to sort of develop biophysics of gene expression of the order of 150 genes. And that's the name of the game, and what we try to do to go to a more complex scenario of multi-genes, being able programming gene expression and generating autonomous systems. Ultimately, the use of surfaces and silicon technology, which we use, is aimed to interface with semiconductor technology, because the surfaces can have electrodes and electric fields and electric field gradients incorporated. We've done something along, and ultimately this will lead to an interface in which the biology communicates with electrical signals. We have started a few years ago something, and we want to pursue that in the near future. Okay, some examples, cell-free gene expression across the scales. So what I'll show you today, not everything, but some examples where we basically go back and forth along the scale axis in terms of both size and gene composition, complexity. I'll show you at the scale of our individual DNA compartment, And what you see here is the red square is fluorescently labeled DNA brush, millions of DNA copies of a given circuit that is programmed to oscillate the expression, in this case GFP. This is, of course, a sped up movie. We typically get a few oscillations overnight. And the reason why you see these oscillations because the compartment is connected to the reservoir and enables us to go up and down in concentration. Otherwise, we have no sync of these proteins. When we couple these compartments in a two-dimensional array, as I'll show you shortly, we generate a tissue-like behavior. It's a multicellular system. And what you're seeing here is a 5 by 5 millimeter chip with about 1,000 compartments organized in a square array. And what you see in these waves are basically synchronization waves that have to do with the coupling and synchronization of all these 1,000 compartments. You'll see where that comes in. If you go down from the millions of copies of DNA and proteins which participate in this kind of smooth oscillatory dynamics of gene expression to the limit of maybe 10 DNA molecules per compartment, and measuring the stochastic expression of individual proteins, as you see here, you can look at, you can look at the fluctuations and see that under decision-making, the dynamics of gene expression looks drastically different than it does at the limit of very large number of molecules. I'll go back to the small scale of a machine, and this is a long-term project in the lab to basically look at ribosome biogenesis in a pure system and try to tackle the problem of new ribosome made from an old ribosome. And for that, we use not only the concept of the DNA brush as a source of gene expression, localized source of gene expression, but also use the rest of the surface to capture the newly made proteins or the newly made ribosomal subunits or other protein complexes. And for that, we use our tailored surfaces. In this case, you can see a line of DNA brushes and an emerging patterns of GFP captured on this hexagonal array pattern on the surface to generate a positive signal when you get both expression as well as assembly and capture on the surface. And last, I don't have time to show this today, but just to sort of show you the direction, we've incorporated electrodes onto these surfaces in close proximity to our DNA brushes and demonstrated that we can actually attract ribosomes or RNA polymerases, in this case in the pure reaction, to basically localize them in the vicinity of a sharp gradient of electric field. This is dielectrophoresis in salty solution at high frequency. And this enabled us basically to show that you can manipulate these gene expression reactions on the surfaces by localized electric fields, thereby opening a new horizon for non-biological control or manipulation of gene expression reactions. I won't show that today, next time. Okay. Yeah. So the basic phenomenology of what we had done, this goes back 10 years now, but just to show the basics, is to create these miniaturized compartments. Again, the scale is about 2 microns deep, 30 microns wide pancakes with capillaries that go around, say, 150 microns or shorter. And this kind of, they all emerge from this kind of microfluidic channels along which we flow in the cell-free expression system, extract or pure. And when you close this from above, each and every one is like a little reactor, which gets materials in and out by diffusion. There is no flow. These are dead-end reactors. So molecules diffuse in and out. And when you start to generate mRNA and protein, they diffuse out. And when they diffuse out, they are carried away at this point. So there's a source and a sink. And when you get a source and a sink in diffusion, you generate a linear gradient of newly made proteins, in this case, GFP. And this is the mechanism by which we have turnover of newly made material in the compartment. When you look at when you can do the math and the physics of this, basically you can map this. You can think of a cell, a small cell, and you ignore any spatial variations within because diffusion is rapid within a cell. And so the governing kinetics is basically that the rate of changes has to do with what you put in the DNA, you program. So if you're just expressing GFP with no regulation, it's a constant. And here is the geometrically controlled effective lifetime of the compartment, which is set by the physical dimensions of the compartment, namely the radius, the width, and the length of the capillary, as defined here, and the diffusion constant of your newly made protein. So this is typically set at the scale of, say, 20 minutes or so. So typically a lifetime which is conveniently, placed to generate steady state concentration profiles or programmable systems which show interesting dynamics like these oscillations. And you can see that just by changing the length of the capillary in this case, you can control the amplitude and the period of the oscillations, everything else being the same. It's the same circuit, the same amount of DNA, etc. And we've demonstrated one-dimensional systems, etc. We studied this, but today I'll show you the most recent work, which, and I forgot to mention, of course, this was initiated by Yale and Alex, back then graduate students in the lab, and they're now independent PIs back at Weizmann. And this recent work is mostly the work of Josh Ricovier, who followed up to basically go from 1D to an entirely new two-dimensional system. And to do that, we basically folded into the third dimension, basically creating these through holes or wells, which run from top to bottom of the wafer, creating a delivery or sort of capillaries like in plants, which deliver nutrients from the underlying flow, which at the backside, where cell extracts or pure, the cell-free expression system basically diffuses in to trigger transcription translation, these reactions, and then the newly made proteins then diffuse out and they're carried away. And so basically we have the same kind of scenario, slightly modified by the well, but that can be taken into account to basically create the same kind of steady state conditions which enable us to program by genes oscillatory behavior, for example. And the analogy here, if you want to sort of go back in history, is the large-scale integration, which basically the first electronic circuitry had to do with about 5 by 5 millimeter chip with about 1,000 components, built monolithically into these semiconductor surfaces. So this is inspired by that large-scale integration in 2 dimensions. And you can program each and every one, and this is how we do so. So this is an image of the chip, and what this spotting instrument does is basically deposit individual drops of DNA into a predetermined pattern. And what I had forgotten to mention is that everything we do is based on a layer which coats these silicon or glassy surfaces to create a sort of an interface between the solid surface and the biomolecules. That interface is based on PEG, which binds on one end to the surface, and the other end it's photocleavable. So by UV photolithography, we can create patterns on which we immobilize various molecules such as DNA, antibodies, proteins, etc. And what I'm showing here is basically a 2-dimensional coupled lattice, a square lattice of compartments. You can see here the resulting DNA before gene expression. So these are dense phases of DNA patterned as a square. The DNA is labeled in red. This is the typical dimension, unit cell of about 150 microns. These black circles are basically these through holes that go into the surface from top to bottom to provide the feeding. And what we show in this movie, which I'm running again here, is individually spotted sources of GFP under no regulation, just to see how individual sources diffuse into empty compartments. And using this kind of an experiment, we basically can calibrate or control to what extent every source communicates to its neighbors. And you can imagine that it decays strongly in space because every compartment dilutes to its neighbor, but also dilutes into the wells in stepwise. So eventually you drop significantly as you go away from each and every source. And this defines an effective diffusion, which is now governed by geometry. And basically nearest neighbor interactions. And so this is like a discrete partial differential equation, which has the rate of what you make of proteins that are made at position I and j are set by the individual lifetime, which is what I showed you for the previous slide. The effective diffusion having to do with coupling between nearest neighbors, and the DNA program that can be different in every compartment or identical if you just look at coupling between oscillators. And what I'll show you now is results which were obtained on this lattice. So what you're seeing here are almost 1,000 coupled oscillators oscillating together in time. And when you look in space, and this is now a rendering of the data into colors, what you can see here is that you generate these waves. You could see it in the first movie I showed you back. And what you see here are that these waves emanate from the edges or boundaries of the lattice. That shouldn't come as a surprise. These are natural defects. Even though everything else is the same, the number of neighbors at the edges or corners differs, and that creates a phase difference, which propagates as a wave. And the extent of propagation, namely, if you look at you can identify traveling fronts in the data. You can do this by looking at the relative phases of the individual oscillators, and look how the phase gradient maps in space. And you can see this beautiful ordering. So when the system is tightly coupled, for example, by having nearest neighbor and next nearest neighbors coupling, so this will be a strong coupling rather than just nearest neighbor coupling, you generate long-range orientational order in this kind of phase gradient diagram, which relates to these fronts, which appear to be rather straight, but of course they're affected by the boundaries, so they fold. But if you take a system which has a sort of a more relaxed coupling, these fronts look more wiggly. They propagate slower, and this can be determined by looking at the power spectrum, namely the amount of amplitude you have here as a function of frequency and wave vector, 1/length scale. And this kind of plot shows you that there's a minimal velocity by which these propagate, and the stronger the coupling, the faster the velocity. And this fits very well with a class of systems that are under the umbrella of reaction diffusion systems that have these propagating fronts that you can model and explain where they come from. Fisher-Kolmogorov, among other class of systems which exhibit these coupled system. I'd like to note that this is the case where you have coupled oscillators, but this is a case study for a more general class of systems in which the same geometry can be implemented with different DNA programs. We don't have to use just oscillators. We can use other systems, and I'll show you in a minute a bistable system. But you can imagine where this goes to, basically to start implementing tissue-like behavior across a scale much larger than individual gene, much larger than the individual compartment, to generate this kind of collective behavior on a scale that is much larger, and you can see it here in this example. What Ferdy Grice in the lab did when he joined the lab a few years back is basically to ask the question, what happens if you program the compartment not by, say, 1 million copies of DNA, but say by 10? Let's drop 4 or 5 orders of magnitude in the number of DNA molecules which participate in the dynamics. And in order to give it a flavor of computation, the simplest form of computation is a decision making. And he used the classic lambda switch, basically reconstituted the lambda switch of the phage. Basically, the basic unit, this is the basic unit. It's classic. I don't need to explain that too much. But the question was, but what Ferdy did then is in order to go through the the transition between left and right, or to actually go through the decision-making point, he introduced a well-known temperature-sensitive mutation in the C1 protein, basically inactivating it above a certain temperature. And that enabled us to basically look at the switch between two states as you go up in temperature. And what Freddie did is study this in the limit of many copies of DNA. And you can see here the resulting dynamics. This is hours. This is expression of GFP, reporting what's happening. And you can see, if you look at the steady state values, that you generate this kind of hysteresis expectedly. Sorry for jumping. I keep moving my cursor. You can see as you go up in temperature, you hit a transition point, and as you go back in temperature, there's hysteresis. This is what you expect in a two-state system. And you can see it because these are steady state compartments. They're open to the environment, and there's a turnover. Otherwise, you couldn't see it in a bulk solution. And now the experiment was keep the same genetic circuit, same switch, but now bring down to about 10 DNA molecules. And you can see, and Freddie was able to basically measure the individual proteins coming from about 10 to 20 DNA molecules without making the correspondence which protein comes from which DNA yet. Nonetheless, in counting the number of molecules, protein molecules being expressed, you could see this erratic and stochastic fluctuations of gene expression. And when he varied the temperature, he could see how the variance in units of the mean actually goes up as you get close to the transition and then goes down. Suggesting indeed that this has to do with occupancy of the various states and fluctuations of occupancy in these complex promoter structure, showing us a drastic difference between slow, precise, and very costly dynamics to one which is fuzzy, fast, imprecise, and cheap in terms of energy. And this is a trade-off that is well known in many computational schemes. Of course, we know that cells basically, use stochastic gene expression. They don't perform computation with 10 to the 5 or 10 to the 6 molecules. It makes no sense. It's very slow, very precise, but very costly. And this is where biology works, basically. So here we have a chance to look at gene expression at the limit of small molecules. And Ferdi wanted to make the linkage between the DNA and the DNA source and the protein, because what came out from this study is that basically we couldn't understand why, despite having few molecules, 10 proteins or 10 DNA molecules in that volume, there was any regulation, because the number, if you look at the concentrations, this shouldn't work. It's just too low. And so the hypothesis that he put forward is that basically what's happening is that you have co-localization of DNA, RNA polymerase ribosome, and the nascent protein. So again, this would be a non-equilibrium consequence. You'll see that in a minute. That was the hypothesis, that there's co-localization of nascent proteins and the respective gene templates. And in order to prove that, he used other reporters than GFP, etc. He introduced the use of the halotag, which, because it acts by a different mechanism, once the protein is made, we made a fusion of a given protein with a given length to the halotag, and the halotag is one that binds this fluorescent dye, which is fluorogenic, and you get fluorescence upon binding. And when you vary the length of the gene, you can generate more and more bursts. And what Ferdi showed for the first time, that you can actually see individual DNA spots labeled. The DNA is labeled in blue, and the protein is in this color, this channel. And you could see bursts of individual, or you can localize the proteins to individual spots and basically look at bursts of a few proteins at a time, measured in between gaps of dozens of minutes. These are typical traces that he was able to prove come from individual DNA molecules that you could see for the first time. The linkage between, and because this was a turf microscopy measurement, you could localize nascent proteins to the DNA at the surface. So you could already localize a nascent protein, but then the next question was, what if you encode on the same DNA a cascaded reaction and from one step to another and actually a full circuit. So I'll jump to the full circuit. And what Ferdi did is basically construct the plasmid, which had two steps, an activator and a repression to close the loop. This thing would typically generate a pulse, and remarkably, what you could see is that the residence times on the DNA of these respective proteins was exceedingly long compared to the stochastic and bursts that you could generate without any circuit. You can really generate these long resident traces of proteins bound to individual DNA molecules. So that was a proof that colocalization actually occurs on a single DNA level. And to make this even more remarkable, Ferdi did this without any confinement, any compartment, actually in a very dilute system with very few DNA on the surface, so there's no communication. He proved that. And nonetheless, this was working, even under flow, which means that the colocalization of the DNA to the nascent proteins actually persists in a cascaded way. And the only reason why this could be explained, because the equilibrium concentration would imply that there's complete unbinding. Everything would favor, and entropy would favor unbinding at this low concentration. And the reason why this works is because this reaction is a non-equilibrium reaction consuming energy, so there could be delays and local concentration gradients of newly made proteins that then bind back to the DNA before they diffuse away. So this is a nice demonstration of colocalization while measuring single DNA, single protein fluctuations. And I won't go into this. This is a new work by Ferdi, basically going from a single plasmid to an entire chromosome. I will just flash it, and for the interest of time, I'll skip it. But just to tell you briefly, he's able to basically take a bacterium, lyse it gently in our DNA compartments, by using these thin capillaries, and basically fill the entire volume of the individual compartment by DNA that can be labeled. I won't go into it. He's seen remarkable things with this, gently lysing the cell and retaining these chromosomes, entire chromosomes functional. He was able to label and see what comes out from the bacteria and subject it to the cell-free bacteria lysate. And in this case, I show one of his results basically to show that he could measure now individual proteins coming out from a single gene in the chromosome. And you can actually look at the stochastic bursts of proteins coming out. I'll leave it at that just to show you that the techniques we were developing enable us now to go from a single gene in the compartment to an entire chromosome or even a synthetic genome that can be incubated or studied in these compartments. I'll shift gear to the remaining of the talk, tell you about our ongoing work towards autonomous biogenesis of the protein-making machinery. The name of the game here is to see if we can find conditions or a scenario for self-regenerating the entire translation system. Our lab has been tackling this for a number of years now. We started with the small ribosomal subunit. We now have results on the large subunit, which is not yet published. I'll show you one slide. And I'll show you also work on the helper proteins or the auxiliary protein, which facilitate translation. Again, remember, I mean, that the challenge here is to take ribosomes and the minimal unit, and the pure, and encode the entire system on genes, and eventually try to see conditions for which you can self-regenerate the entire genetic system, the ribosome plus all its auxiliary proteins. And of course, it's tough to do everything in one shot. And I'm not going to review the literature. Other groups, of course, are tackling this in various ways. I'll show you our own work in the interest of time without a review here. And this, I'll start by actually the most recent work, and I'll focus on the translation helper or auxiliary proteins, which are sort of schematized here. I'll start with that because the concept introduced here would be going using the surface, the DNA brush, and the capture on the surface. That will also lead us to what we had done earlier to talk about how we build ribosomal subunits. So briefly, without going into any detail, this is the sort of biochemical scheme of all the helper proteins that help the ribosome translate from, okay, I'm trying to, yeah. So initiation, amino acylation, elongation, and termination. You have 30 proteins, 30 genes, which are encode them, initiation, etc. 20 synthetases, elongation, and termination proteins. They have different functionality. I'm not going to go into that. The idea is basically to demonstrate that you can now self-regenerate each and every component and then put them all together and see if you can self-regenerate all of them together. And so this was done in 2 steps. The first one was in solution, and this is an experiment with basically kind of simple, but it took a while to figure out how to do. And the idea is the following. You take all the pure proteins, and now you remove one of them, and you lose the function. And you lose the function even if you add the gene coding for that protein. This is a typical example of one of the pure proteins. Now you take the same one and then you add a small amount of seed, so purified protein, just enough such that without adding the gene, you still see no recovery. But if you take the seed, and the seed could be 1% or 10%, depending on the protein, but you add the gene coding for the protein, now have recovery. As you can see, it takes time. This is an autocatalytic, if you want, reaction, self-synthesis. It takes a while to come out, but eventually it does. And so this is the recovery assay, and we did that for each and every one of the pure proteins, one at a time. This was done together, of course, with the inventor of the pure, Shimizu, Yoshihiro Shimizu from Riken in Japan. This work is with collaboration with him. And the next step, I'm cutting short, many experiments and so on, but the idea I want to show here is this sort of catalytic idea. This is our DNA brush, which now codes for all the respective genes, 30 of them. And this would be, for example, the composition of the DNA brush, which we encoded. So it has all the genes, factors, initiation, elongation, release, synthetases, a reporter gene. This would be GFP, and the GFP has a tag, an HA tag, for which there are antibodies patterns nearby. So think of a little fountain of, so you have the DNA brush is being expressed, the genes are expressed, the proteins are made, and the reporter gene comes out, and then it, like a fountain, is captured nearby to give us these green patterns, in this case, hexagonal patterns when it happens. And the name of the game here was to ask the following question. Now that we have the individual seeding assay or recovery assay I just showed you, what happens if you generate, you put them all together, you bring it down to the bare minimum that it doesn't work, and lo and behold, when you now add the respective genes and you increase, you put them all together in the DNA brush, we found conditions where basically it doesn't work unless you put them together. So you can wait and wait and wait, and it takes 4 to 6 hours to start seeing GFP coming out. But this is the first sort of self-regeneration of all of them together as a whole system happening simply because you generate this kind of local feedback in which, again, non-equilibrium reaction, everything is very, very dilute. So this wouldn't happen in equilibrium. And this kind of scenario, dynamic scenario, as we portray it here, has this kind of local feedback. So newly made proteins remain long enough there to basically jump back into the DNA brush, get into the pool of available proteins and start to create feedback. And all you need is a small seed of some of the proteins to basically activate the self-synthesis of the entire translation auxiliary, all the translation proteins all put together. So this is an idea that we implement in 2D, but of course, the principle is general. What's required here is a seeding, the idea of seeding. So you reduce the concentration to the bare minimum, and then you add the genes. And the secret sauce, if you want, is that you basically reconstitute the confinement, the localization, everything works together because locally on the surface you have high concentration, but only locally. The global concentration of these newly proteins is ridiculously low. It wouldn't work otherwise. So you can think of realizing this in a vesicle, in a membrane compartment, or otherwise. Now, the same idea goes to basically making a ribosomal subunit, and here is earlier work. This is mostly work by Michael Levy, who is now a PI at the Technion, and Benny Falkovich, who's graduated at MIT. Noah Vidal is a graduate student who now mostly work on the large subunits. I'll show you this in a couple of slides, and that will be sort of the end. These papers basically show the tool development. The actual paper published 4 years ago, basically showing the idea is the following. So you express now the proteins and the RNA of the small subunit of the ribosome. And you make the RNA and you make the proteins, and you need to capture them on the surface. And what we use is, so this is the annotation of this is the structure of the small subunit. And here's what we do. The trick is to introduce a fluorescent probe into a broccoli aptamer, which is inserted into one of the available loops that can accommodate into the ribosomal RNA of the small subunit. And there are 20 respective proteins which bind the ribosomal RNA, fold it into its stable structure in a hierarchical manner. And this is the well-known Nomura plot, which demonstrates the equilibrium map discovered some years back by Nomura, basically showing that there's a hierarchy of primary, secondary, and tertiary proteins that create this kind of dependency map. You first need to bind the primaries in order to bind the secondary and the tertiary, depending on the 5' middle or 3' regions of the ribosomal RNA. And we need to generate a signal and measure it by turf microscopy. What we've done is basically fused one of the ribosomal proteins to the HA tag, for which there's an antibody on the surface. And so the signal you see on the surface appears only and if and only if two things happen. Both the ribosomal subunit is brought to the surface, so the RNA, which carries the fluorescent signal, is brought as a complex to the surface. And we do it for each and every one of the respective ribosomal proteins to generate an entire projection of the kinetics of binding. And I show you here just a final result of all the 20 bodies, so to speak, 20 proteins encoded in the DNA brush. And each and every one generates a curve. Each one is an experiment done with all the proteins in RNA, so it's identical, but under a different one, bringing back the subunit to the surface. This experiment runs in parallel, 20 different reaction centers on the surface. It's a glass surface in this case, and measured by turf microscopy simultaneously. And you can see here there's a timeline. If you look at the onset of assembly and plot it as a function of time, you can see how the individual proteins enter into the growing assembly, and there's clearly a match, which is verified by other experiments like deletion and two-body experiments. So the advantage is that you can program things as you wish, from two-body interactions, one protein and RNA, to deletions, to groups, etc., you name it, and we've done some of it to generate this kind of kinetic map of interaction of the subunit assembly in vitro. As you can see, it's stretched into long times. This, goes all the way to more than an hour until you see some of them coming in. This is much slower than what you would expect to see in the live bacteria. Live bacteria, obviously, this is a much longer length scale. There is diffusion over many hundreds of microns eventually. So this time is stretched and the measurements is limited. but we now have a more confined realization of this that hopefully would give us even faster kinetics. Remains to be seen. I'll just show you that the same principle works for the large subunit. This is work by Noah Vidan, who basically has been tackling the large ribosomal subunit, which is far more complex in structure and the number of protein. It has chaperones and RNA modifying enzymes, which are not part of this specific result I'm showing yet. So what this map shows you is, again, a dependency map, which is far more complex than the large subunit. We now know that this is far more plastic dynamic than anything else. It's not a one-to-one kind of correspondence, and it's mostly annotated by various domains which form in time. And what Noah has done is use the same methodology of introducing broccoli aptamers into various positions of the large subunit ribosomal RNA, and use the same trick of expressing proteins and capturing each and every subunit by a different protein via the linkage. And here I show you that these are kinetic traces. You can see there's a lot of information. Some of them emerge early, and some of them are stable, some of them are less stable. But what's clearly showed is that basically you recapitulate the large scale picture, the big scale picture of having the first layer basically lighting up first, and actually very, very little response in this specific experiments when you have the late stages, suggesting that some of the the factors and modifying enzymes are essential for further assembly and functionality of the large subunit as expected. But we recapitulated the essential parts of the assembly, even of the large subunit. Okay, so I'm going to end here and show you this kind of a summarizing slide. We have basically developed a program which uses our DNA compartments to basically look at cell-free gene expression outside the living cell with increasing complexity. And complexity could be the number of genes, and whether we look at a circuit with a few components or a machine with many components, whether we look at a single DNA and single protein expression kinetics or under a circuit, or whether we go to the sort of limit of a tissue-like behavior of, in this case, 1,000 cells. I didn't show you work that we did a few years ago to look at the E-field manipulation in the absence of time, but you can imagine that by looking at these reactions under forces which are gently applying electric field gradients to attract ribosomes, as I show here, basically this could be used to manipulate the rates of protein synthesis or RNA transcription in these reactions, because the DNA is localized, and this gives us a handle on these kind of reactions. And ultimately this can lead to a different kind of system where the artificial cell, quote unquote, is now being regulated or manipulated externally to interact with the environment, not only by biological parts. I'll finish up by thanking some of the people I mentioned in this group and our current group, with various people who've left the lab and people with whom we collaborate regularly. Vincent Moirot, our my long-term friend and collaborator. We started together as postdocs. We still work together continuously, and Yoshihiro Shimizu from Japan. And that's it. Thank you. I can take some questions.
Thank you so much. That was awesome. There is quite a bit of questions in chat. Can you see chat?
I'll look at it now. Yeah.
If you could read a question out loud before answering, because people watching the recording don't see the chat.
Okay. Okay. Do you think proper artificial cell has to be membrane bound, or can it be compartmentalized in some other way? Okay, so I alluded to that. I mean, this is a decision. Let's call it embodiment. Maybe there should be a better word for what we do. Maybe DNA compartments rather than artificial cell. To me, an artificial cell is anything but what life has found. So it doesn't have to be membrane bound. but that's okay. I mean, if you don't think of this as something which necessarily has to divide, then we're creating a system which can express, can maintain homeostasis. Ultimately, we think this is an easier path to show self-regeneration in steady state, and leaving aside the problems that have to do with division and coupling everything together, the division, exchange of materials, physically separating daughter cells. This is ultimately the goal if you think about real minimal cells, bottom up, etc., building a cell. But we're proposing that using these DNA compartments, you can study principles that are not specific to what we do, but could be generalized. So the idea of creating reaction centers, the DNA brush idea of putting many genes together at close proximity is essentially a physical or a spatial operon. And you can think of it as sort of chopping an entire genome into small pieces next together. But the colocalization idea would transcend most likely to a full, contiguous molecule as a genome. The colocalization and the consequences of being out of equilibrium to generate local patterns or global patterns is something that would transcend as well. So for me, the use of 2D compartments is just a way, the technique to study the fundamentals of gene expression reactions under confinement, and what are the considerations to build complexity bottom up. So proper artificial cell remain, as a definition. Okay. Thank you for a great talk. Thank you. What is the maximum size of the capillary in your solid state compartment? Basically, how many reactions together can fit into a single compartment? Very good question. We are now working on answering that question in various ways, either by doing multiple genes, which are separate circuits in one compartment, or coupling compartments in space. Different reactions than I showed today. Today I showed you coupled oscillators. We've also demonstrated the bistable switch. But the question is correct. How many different reactions we can put together in a single compartment is a very good question. It has to do with understanding resource sharing and encoding capacity, etc. So this is something that we're still working on. Okay, why is computing with lots of molecules slow? Aha, surely at the same average concentration, it's roughly the same rate. I agree that it would be costly. Okay, so obviously when you're doing solution experiments, you're averaging out many individual experiments, and you cannot decipher, look at the individual DNA doing the same computation, obviously. So that's why there's a smear. It's slow because you're looking at an ensemble average in a test tube reaction with many, or in this case in a compartment, everything happened, it's not synchronized. So you don't see the fluctuations. When you go down to the zoom into each and every DNA molecule, you see the stochasticity. So of course, nothing changes per se. It's the same reaction. We're just looking at it one molecule at a time and identifying the individual stochastic or footprint of individual molecules. But if you average them out, you will see these smooth things become slow. And costly, yes, indeed. I mean, the nice thing is that you see fuzzy, sort of the fluctuations demonstrate a principle of fuzzy computation, things which are not precise. That's the point I was trying to make. It's not necessarily on and off. It fluctuates, and you need to, see how the fluctuations correspond, in this case, to the topology of the circuit, being a bistable switch with two repressors. This is where the fun was, to basically identify that this kind of fluctuations actually correspond to the circuit underlying. And the nice thing, it's not a cell, it's not a living cell. People did that 20 years ago with looking at circuits at a small number. But of course, you have a cell, so many things are happening simultaneously. Here, you look at just one circuit or one component at a time. So this is the advantage of doing things purely in vitro. Okay, interesting story about colocalization. How exactly do you think pushing the system out of equilibrium encourages rebinding of the newly produced proteins? It's a consequence. We're not pushing the system out of equilibrium. It is out of equilibrium, very far from it. Transcription NTPs are the currency, and ATP and GTP is for translation. So we are driving, in order to do polymerization reactions of RNA, making RNA and protein, the system has to be out of equilibrium. It is. And so it's the colocalization is a consequence. Simply said, the physical picture is that you have the RNA latched to the DNA to make RNA. The RNA, we know this, this has been very validated for years, in vitro, as well as, bringing things outside of the cell, that there is colocalization. DNA, RNA, DNA, RNA polymerase, RNA, and then ribosome and protein. The question was, does it stay long enough, linger around long enough near the same DNA to basically hop over before it diffuses away? And the answer was, yes, it does. Not all the molecules, but a fraction do that. So it's a consequence. It's not something that you push. It's a measurement that is an observation. Okay. Awesome work. Thank you. Would it be possible to add DNA replication, for example, rolling circuit polymerase like the 529 that Danilol uses, and have the polymerase replicate genes from the DNA brush? Yes, we are thinking about, we haven't yet done DNA replication. We've done other reactions like DNA recombination. And we're now thinking of doing DNA replication either from the brush or from other constructs in the compartment. What is the yield of ribosomes produced in the system? How well do the ribosomes produced on the wafer functions compared to other ribosomes reconstitute ISAT, R-ISAT, wild-type E. coli? General answer will suffice, but I'd like to get precise answer eventually too. Okay, fair enough. The experiments I showed you were done in a sort of semi-open system where we don't contain all the newly made proteins and assembled ribosome subunits. Because this was sort of a semi-open surface, we couldn't measure the yield of ribosome production. We are now doing so in confined compartments to be enabled exactly to answer this question: what's the yield of ribosome subunit assembly in a case study where we can measure that? I need to mention, based on your question, which I didn't emphasize, and thank you, that these ribosomes have not yet been functional. We synthesized the small subunit. We demonstrated that it can bind to a purified large subunit, but we haven't yet been able, certainly not in this pure system, to basically demonstrate a new functional ribosome. That will be a big step forward, especially in the pure system, because you have to include all the genes, which would enable to generate an entire functional ribosome, namely RNA modifying enzymes and special factors, which chaperones that or helper for proteins that fold the ribosomal subunits properly. So that's work in progress. Could you use TXTL instead of pure? Wouldn't the yields be higher? Sure, we're trying to do that now. Thank you. Yeah, of course. And there's a report by the Japanese group recently that have demonstrated an entire ribosome in the extracts, in the TXT instead of pure. Yes, follow up. Let's follow up that work. What were the minimal set of RNA modifying enzymes you need to add for? That's a great question. We're studying that right now. What I showed you now has no use of no introduction of modifying enzymes. Therefore, we didn't expect anything except for initial stages of assembly. That was the first step. What I showed you is just the first step of what eventually, if we succeed, or if one succeed, is to include the modifying enzymes as well as the chaperones. So far, no, not yet. Have you tried producing ribosomes from other species? Aha. We have done some work on a different ribosomal subunit. It's not yet published. We're working on it. It's an interesting study where basically you can program, you can use the pure system of E. coli to generate a small subunit of another bacteria. And that's something we did. It's not yet published, and there's interesting stuff there. Yes, so we are doing that. Have you tried minimizing the ribosomes? Reducing the amount of ribosomal RNA, ribosomal proteins. Very good questions. The answer is not yet. what would you need the concentration rate of ribosome production to be in order to remake all proteins in pure using your wafer system? Very good question. So, okay, guys, this is very challenging. Everything I show you now, 90% fails and then some of it works. So all these questions are in the pipeline to see exactly what's the concentration and what are the conditions to generate these scenarios. Generally, I'd say that we found that by reducing the overall pure concentration to the bare minimum generates the strongest contrast at the price of taking much longer time to self regenerate until after 6 hours in a pure reaction, which typically dies out after an hour. It takes 6 hours on the surface with the sensitive turf measurement to start seeing GFP measured after so many hours under this self-regenerating loop that we introduced in the DNA brush. I hope I've answered some of the questions. That's all I have for now listed.
Thank you. Thank you so much for answering all the questions. I think that was basically a second talk, and that was awesome. Yeah, I'm really looking forward to the self-regenerating ribosome, because I think that's really going to be a game changer once you have a functional self-regeneration.
Of course, yeah, that's the holy grail, one of a few holy grails. I agree with you, and that's it's a worthy effort, and our approach is by far not the only one. Some of the principles could be implemented by other ideas, other realizations. Again, the DNA brush is just a trick to enable this localization and measurement. You could imagine different scenarios that would basically establish a similar scenario. Okay.
Thank you so much. And
Somebody has
last question, yes.
You have another question. For the recovery assay, remove one protein. The graphs you show don't reach the full. Yes. Does that maximum? Okay, so that definitely is the case. Does the maximum translation capacity change as you change the amount of seed protein? Of course. It's extremely, that bottleneck where you remove basically the protein and you add increasing amounts, you basically create a bottleneck where it's necessary. So it's ultimately extremely sensitive to the amount of seed proteins until you hit a limit where it's basically less sensitive, and then you recover by adding the DNA. This is exactly the idea. Yeah. Okay.
All right. Thank you so much.
Thank you.
We are at time. And I want to say thank you, Roy, so much again. And thanks everyone for a great discussion. And see you.
Okay.
See you soon. See you later. Bye.